Okay, well, good morning everyone. We'll make a start. I hope you had a good weekend and back for another week of learning in your studies. Now, what we've been looking at in this block of lectures is this phenomenon of oxidative stress, and we've seen how that during normal respiratory function in aerobic cells we get the release of reactive oxygen species, including some pretty nasty free radicals that can attack cellular components. In particular, last week we emphasised some genetic damage associated with free radical formation, and then we looked at, in Thursday's lecture, protein damage caused by oxidative stress. Now, I've left as a third target for intracellular damage lipids because in some ways this is perhaps the most important, in the sense that it generates some pretty nasty products that can act as mediators of oxidative cell damage. And so, whereas a lot of free radicals don't have the ability to travel too far in cells and tissues, some of these noxious products of lipid breakdown are quite diffusible, and so it's very likely that they are causing and contributing to cell damage during oxidative stress. So this particular lecture has been timed in a way to help you with your labs you're doing this week. We're going to be looking at whether those dozen or so compounds in our compound library, how good they are at protecting cells against aldehydes formed during oxidative damage to cell membranes. So hopefully today's lecture will help you make sense of what's going on in the labs this week. Okay, so what is this phenomenon that we're interested in this week then? It's lipid peroxidation, and this is a phenomenon that was really first identified by free radical chemists in the rubber industry, and it was a while before people realised that it also happened in cells and tissues and had medical significance. But people in the rubber industry realised that, of course, rubber products have a finite lifetime. If you've ever had perhaps a bike that belonged to your dad or something, left it in the garden shed for a couple of decades, you'll find that the tyres go white, and the wall of the tyre often becomes a bit fragile and vulnerable to rupture. And that's a consequence of oxidative damage to some of the lipids in the wall of the tyre. So the basic phenomena involved in lipid peroxidation was worked out by these chemists trying to figure out ways to stop this process of rubber deterioration and improve the shelf life of tyres. So these days modern tyres have significantly longer lifetimes because they put a lot of antioxidants in there to preserve the rubber. Now, it's also important in food chemistry. So again, it's not something we see a lot necessarily these days, but in days gone by, you would often have a tub of fat in the kitchen, and sometimes if you weren't careful in using it, it would go rancid and white, more solid, solidified, and very smelly. And that's because of the release of volatile substances during this process of non-enzymatic breakdown of lipids in fat. So this process is a determinant of the shelf life of processed foods. So that's one reason why food producers put synthetic antioxidants and naturally occurring antioxidants in food, just to make them have a longer shelf life, so that we can ship them all around the world to different markets. One of the things that allows us to do that is the presence of antioxidants. But of course, we're really interested in the role of this process in cell damage and tissue ageing, and really the current interest in lipid peroxidation really stems from the discovery, really back in about the 1990s, I think, that a lot of the products formed from lipid peroxidation are themselves toxic. And so whenever oxidative stress occurs in a tissue, we can get membrane damage, releasing some pretty nasty substances that amplify cell injury. Okay, so why is this phenomenon a problem? And it's really got to do with whether or not a particular populations of fats contain polyunsaturated fatty acids. So lipid chemistry gets pretty complicated. I've just put a fairly simple introduction here in these structures here, but of course lipids are essentially long chain fatty acids with long aliphatic side chains that make them nice and greasy. And we can distinguish between different fatty acids, firstly in terms of the number of carbons in the chain. So one of the important biological fatty acids there at the top is stearic acid, and as you can see, it's got 18 carbons, and but no double bonds. So that's an example of what we call a saturated fatty acid. And then if we introduced a single double bond, then we have a leic acid, the same number of carbon atoms, one double bond. And this is, instead of a saturated fatty acid, we call this a monounsaturated. And then if we introduce more double bonds, we enter the realm of polyunsaturated fatty acids. And so you can see with 18 carbon fatty acids, if it's got 2 double bonds, we have linoleic acid. Linolenic acid has 3 double bonds. Add another couple of carbons and another double bond or 2, and we get to arachidonic acid, which has 20 carbons and 4 double bonds. And of course, that's enormously important in pharmacology because it's a precursor to prostaglandins and leukotrienes and some very important physiological and pathological mediators. So arachidonic acid, very important in pharmacology. But then if we keep going and adding more double bonds, we can get to eicosapentaenoic acid, EPA, which is present in the fish oil we're going to be using in our experiments this week. And then also, it's also rich in 22,6, docosahexaenoic acid. So that contains 22 carbons and 6 double bonds. So now, why is this important? Well, the more adjacent double bonds a molecule has, the more vulnerable it is to free radical damage. And this has got to do with the energy that's required to break a carbon-hydrogen bond. So we look at that in a, when the carbon-hydrogen bond, so this is, remember that's what a hydroxyl radical does. It loves to steal hydrogen atoms off other molecules. It's extremely reactive, that's what it does. So when it encounters a lipid, it's going to take the hydrogen from the carbon-hydrogen bond that's the easiest to break. That's the basic idea here. Now, if that methylene group is adjacent to 2 saturated carbons on either side, then it takes a fair bit of energy to break that bond. But if there's a double bond on one side of the methylene group, CH2, this is just a methylene group, just any old carbon here. If it's in a fatty acid, if it's adjacent to a saturated bond on one side of the carbon and on the other side there's a double bond, an allyl group we call that an allylic methylene, then the energy required to break the carbon-hydrogen bond drops by about 12%. But then if there's a double bond on either side, we call this a bisallylic methylene group. Bis means two, so there's an allylic group on either side of the methylene. See how the energy required to break that bond drops considerably by over 20-25%. So if a hydroxyl radical attacks a membrane, it's going to preferentially remove the attack of polyunsaturated fatty acid because there's just less energy required to get that process of oxidation occurring. Okay, so that means that some of these fatty acids that contain lots or the presence of a bisallylic group, so that is a double bond flanked by methylene flanked on both sides by a double bond. You can see linoleic acid's got one, linolenic acid's got a couple, arachidonic acid's got 1, 2, 3, and so on. So the more double bonds a fatty acid has, the more bisallylic methylene groups, the more vulnerable it is to free radical damage. So the upshot of this is that when you take an animal and you subject them to free radical exposure, classic example will be a compound called carbon tetrachloride, we'll talk about in a minute, causes strong lipid peroxidation in the liver. If you extract the liver and then extract the lipids out of that tissue of that animal, it's undergoing oxidative stress, you often find selective depletion of these polyunsaturated fatty acids while the levels of monounsaturated fatty acids and saturated fatty acids don't change. Okay, so there's a selective consumption of polyunsaturated fatty acids during this process of free radical attack. You probably all know this, but just a couple of basic terminology points that we in regards to lipid naming lipids. You probably heard about omega-3 and omega-6, or N6 or N3 fatty acids. Those two terms, omega and N, are basically interchangeable, but essentially the omega carbon is the N carbon in the fatty acids. So the 6 and the 3 is telling you where is the first double bond located in that fatty acid relative to the N carbon. So if it's the first double bond in the case of arachidonic acid, you have to go in 6 carbons from the N to get to the first double bond. It's an N6 fatty acid. Same with linoleic acid, you've got to come in 6 carbons before you encounter the first double bond. Whereas linolenic acid, the first double bond's on the 3rd carbon. So that's an N3 fatty acid or an omega-3 fatty acid. So you'll probably hear those terms used quite a lot in health contexts these days, because people look at the implications of health implications and of consuming different types of lipids in your margarine or whatever. Okay, so that's the sort of lipids that are vulnerable to free radical damage. Now we want to look at the actual process and see why this phenomena is so nasty in terms of its toxicological implications. So the people in the rubber industry, the chemists, were quick to work out that there are several different stages or phases to oxidative damage to lipids. And the first phase they identified is the initiation phase, and we've sort of talked about that in a way in previous lectures. This is where something like a hydroxyl radical usually attacks a lipid, which we've just designated here as LH, and let's assume it's a polyunsaturated fatty acid, and it basically snaffles that hydrogen and one electron only. Remember that hydrogens are attached covalently via sharing of 2 electrons between adjacent carbon and hydrogen atoms, and the hydroxyl radical attacks and abstracts one of those hydrogens and leaves the other electron on the lipid. So this is a lipid radical. It's the initial product of an initiation reaction. So we call that process initiation. And these carbon-centered radicals, because this free radical is on a carbon atom, are very unstable chemically, and they rapidly react with oxygen to form what we call a peroxyl radical, or lipid peroxyl radical. Now, these are nasty species because while they're not as chemically reactive as hydroxyl radicals, they are very lipophilic and greasy, so they can diffuse through cell membranes, and they're very reactive with other lipids, so they're actually good at carrying out that same sort of hydrogen abstraction reaction themselves. So this is why they're so damaging, because they can amplify free radical damage to membranes. And this reaction is known as propagation. So here we see a peroxyl radical doing this sort of attack on an adjacent lipid. So here we've got a unoxidised normal lipid, LH, being undergoing hydrogen abstraction by a peroxyl radical, and it's generating what we call a lipid hydroperoxide, LOOH. But then see how it's also generated a new peroxyl radical. As a result of the formation of a lipid-centred radical, then trapping, or carbon-centred radical trapping oxygen to form a new lipid peroxyl radical. So now we've got two oxidised lipids where we previously had one. So, and now this peroxyl radical is free to start going off and attacking another lipid. But also the lipid hydroperoxide, if there's any iron around, it undergoes oxidative reactions with iron and forms more peroxyl radicals and alkoxyl radicals. So I'm struggling with this mouse here today. But alkoxyl radicals, it's LO dot. And so we're now getting multiple free radicals being formed from one initial damaging event by a hydroxyl radical on a single lipid. Now it's sort of cascading through the lipid membrane. So you see why this is so nasty and damaging to cells, because we're getting multiple lipids formed from an initial single initiation event. Okay, so cells go to a lot of trouble to detoxify lipid hydroperoxides, things like glutathione peroxidase, a very important enzyme for detoxifying lipid hydroperoxides. It's part of the antioxidant defences of cells to try and keep this chemistry from happening. But then also another way cells try to counteract this process is by accumulating vitamin E, alpha tocopherol, within the cell membrane. We'll come back to that in a minute. Okay, so the third phase of lipid peroxidation, identified by those rubber chemists years ago, is termination. And this is where we get reactions occurring between free radical species that form non-radicals. So in a sense, it brings the cascade reaction, or at least a particular pathway, to an end. And one of the things that can happen to cause this outcome is the formation of cross-linked lipids. So here we've got two adjacent lipids reacting with each other, forming a new stable product called, with a covalent bond between two adjacent lipids. Okay, so this is not that good an outcome, because the whole point of a lipid membrane is it's got to be flexible and bendy and able to cope with stresses. So if you're getting permanent covalent bonds between lipids, then the cell membrane is going to lose a bit of that fluidity and ability to function. Okay, so this is one of the things that happens as you get older, unfortunately. The number of cross-linked lipids in the membranes of our tissues tends to increase. And it's just reflecting the accumulation of oxidative damage of this kind due to cross-linking. But another really important way of terminating lipid peroxidation is for antioxidant such as alpha tocopherol, and it donates, so that's vitamin E. Vitamin E is very good at blocking lipid peroxidation, and so it donates a hydrogen to the lipid radical to regenerate that intact non-oxidised lipid. And in the process, vitamin E forms its own radical, alpha tocopheroxyl radicals, We'll talk about this in my lecture on antioxidants, but this is not a very reactive radical, sort of a low reactivity radical, so it doesn't attack lipids. So this is why this is a detoxification, and then it gets regenerated by ascorbate and glutathiones, so in the cytosol. So cells have got very good capabilities for detoxifying these lipid radicals via vitamin E. Now, another thing that happens during the termination phase, and I haven't shown it on this diagram, is you get the formation of some of these oxidised lipid fragments, and these are aldehydes in the form of malondialdehyde. And this is what we're going to be measuring in our labs this week. You'll be setting up your lipid peroxidation chemistry, letting it rip for an hour in the presence of your suspected antioxidant compounds from your library, and we'll be measuring the levels of malondialdehyde as a product of lipid peroxidation, just to tell us how good your antioxidant is at blocking this damage. Now, okay, so very early on, when people began studying this phenomenon of lipid peroxidation, they tended to think that the main consequence was for the membrane itself, that this oxidative process might be damaging membranes, and if you got enough of it, then you might get leaky membranes to the point that they can't perform their basic membrane function of controlling the movement of molecules in and out of cells. And so it's one of the basic functions of a membrane, plasma membrane in particular, is to keep everything in the cell, inside the cell. And so when cells start dying, their membranes start rupturing, necrotic type form of cell death. Okay, so people used to think, well, for lipid peroxidation to be toxicologically relevant, then it would have to trigger membrane damage, loss of control of cell contents for it to be pathologically relevant. The problem is with this theory, and for a long time people sort of gave up on lipid peroxidation in a way, because this sort of overt membrane damage, this sort of necrotic cell death in the form of releasing cellular contents and rupturing, usually occurs fairly late in pathological time frames. And so people realised that lipid peroxidation, or at least concluded that lipid peroxidation must be a fairly late stage event in tissue pathology, and it wasn't of that much importance. Right? It's just something that happens in already dying cells. Something else must be causing the cell to die, and then it ruptures and undergoes membrane loss, membrane damage. But then, like I said, in the late 80s and 90s, another theory began emerging when people realised that lipid peroxidation was actually generating toxic products. The oxidised fragments, or fragments of oxidised lipids, that big long alkyl chain gets oxidised and breaks up, and it releases all these oxidised species, including aldehydes. And that these are capable of causing damage to cellular components. And this is this sort of rejuvenated the lipid peroxidation field, because now people had things to measure in terms of damaged products and to sort of incriminate this process in pathology. You always need good biomarkers, and so this just gave us a way to confirm the presence of lipid peroxidation in a tissue in the earlier stages of disease. Now, for the rest of the lecture then, I just want to have a quick superficial look at some of the species that are formed during lipid peroxidation, and we'll see why some of them are of toxicological interest. And I want to classify them really on that basis. How significant are these species in terms of disease mediators? So I'm going to focus on that newer idea that lipid peroxidation generates toxic substances that amplify cellular injury. Okay, so when you look at lipid peroxidation products, and it literally produces hundreds of different chemical substances, as you can imagine, if you've got a lipid membrane containing a whole variety of different lipids, then if you trigger lipid peroxidation, you're going to get lots of products being formed. Some of them are likely toxicologically relevant, others not so much. And so I want to just firstly look at some of the low toxic potential products, and this is really because they They don't have very high chemical reactivity, and this includes things like alkanes. But nevertheless, even if they're not that chemically reactive, they might still be useful biomarkers for telling us how much lipid peroxidation is occurring in a tissue. But because I'm a chemical toxicologist, I'm obviously more interested in products of lipid peroxidation that do have chemical reactivity and toxic potential. And when we look at those, we can subclassify them according to free radical products. So that includes some of those peroxyl radicals and alkoxyl radicals. But especially important, I think, are the aldehydes, the non-radical products. And these can be very electrophilic and reactive with cellular components. So I think they deserve particular attention as mediators of disease. Okay, so let's start off with a couple of classes of substances that are released during lipid peroxidation, and starting off with those that have low toxic potential. Now, the alkanes are perhaps some of the oldest recognised products of lipid peroxidation, and these are just very small molecules, very small hydrocarbons. Now, you would have learnt about these probably in your very earliest organic chemistry classes way back in high school. I don't know when you do organic chem in high school these days, probably year 9 or year 8. You probably learnt about these very simple hydrocarbon molecules. But they're very gaseous, they're very volatile, and these are actually one of the reasons why you can smell advanced lipid peroxidation in that rancid fat that I mentioned earlier on. If you have a tub of cooking lipids that's gone off and oxidised, you can often smell the organic volatile substances, and you're mainly smelling some of these alkanes. Okay, so they form via breakdown or scission, cleavage of that long hydrocarbon tail of the polyunsaturated fatty acid. And one of the interesting things about them is that people have used them as whole body products of lipid peroxidation, because you can imagine, because they're so volatile, if they're formed in the body's tissues, they because of their low vapour pressure, they get exhaled in our breath. So you can measure concentrations of these substances being exhaled from in people's breath. It can tell you how much lipid peroxidation is going on. Not a very sensitive marker, but it can be a potential indicator of how much lipid peroxidation is occurring in the whole body. Now, I mentioned earlier that one of the chemicals that's had a lot of impact in the lipid peroxidation field is carbon tetrachloride, and enormous amount of research on this molecule. It's often used as a tool to induce selective lipid peroxidation in the liver. And what goes on with this molecule is that it undergoes oxidative metabolism by cytochrome P452E1. So carbon tetrachloride, as the name suggests, very simple molecule, just one carbon with 4 chlorines attached to it. Pretty stable chemically, but it does undergo oxidation by cytochrome P450, and it breaks one of those chlorine carbon bonds and generates what we call a trichloromethyl radical. So it's a bit like a lipid radical formed during lipid peroxidation. Because it's unstable, it wants to react with oxygen. So it forms this peroxyl radical, trichloromethyl peroxyl radical. So this is a nasty species, and it's very damaging with lipid membranes. So remember that P450 is located in endoplasmic reticular membranes. So when you get metabolism of carbon tetrachloride, you're generating lots of this nasty free radical in the directly in the lipid environment of the liver. So we get lots of lipid peroxidation in the liver. And this is toxicologically relevant because once upon a time, carbon tet was used in huge quantities in the dry cleaning industry. It's also used a lot in chem labs. I don't know if it's still used in chem labs. I don't know if anyone is doing organic chemistry synthesis major, but when I was a student, we still used carbon tet because it doesn't have a proton in it, so you can dissolve molecules in it and put it in an NMR tube, which monitors proton signals. And so you can just look at your molecule without any complicating effect of the solvent. But it's probably been banned for that purpose. You might still be able to use it, I'm not sure, but in a chem lab, but you certainly can't use it in dry cleaning industry because people found that there is a higher incidence of liver disease and also liver cancer associated with people working in the dry cleaning industry. Okay, so the reason it's so popular in the dry cleaning industry is that it's a very good solvent, so it dissolves all the gunk and dirt and that in people's clothing, and gets them nice and clean. But of course, it was having effects on the people working in that industry. Now, but because of when people realised that this is such a strong inducer of lipid peroxidation, the discovery really goes back to the mid-70s, and you can see where what people have done. They've made liver homogenates from control and carbon tetrachloride treated rats, and what they've shown is that in the control rats, livers, very little ethane release, so ethane being that simple 2-carbon alkane produced from lipid peroxidation. And you can see that in the carbon tetrachloride animals, the liver's homogenates were churning out the ethane. And if you gave the carbon tet to an animal with a P450 inducer, remember I talked about that in first semester, you can switch on the expression of P450s in the liver, increase their concentration. And so if you did that with phenobarbital, strong P450 inducer, you increase the severity of liver damage, but also the yield of ethane in these livers. And then if you administered the animals with vitamin E, alpha tocopherol, you're able to reduce the degree of ethane release. So it's just an example of the sort of things people used to do in this field. Not so common these days because you have to use fairly complicated systems, closed tubes, and extract the volatile products of the reaction from your test tube, and it's just a bit fiddly. So now we have other ways of monitoring lipid peroxidation. Now, a big deal happened in about the the late 80s or early 90s with the discovery of the isoprostanes, and this was quite a big deal. It actually was, a lot of it was done in pharmacology department at Vanderbilt, where I did my postdoc. I did it in biochemistry and toxicology, but nearby they were doing this exciting research on isoprostanes. And isoprostanes are basically products of non-enzymatic free radical attack on lipids. So hopefully you've learned about prostaglandins in other lectures in pharmacology, really, really important mediators of a whole lot of physiological responses. They're products of the cyclooxygenase dependent metabolism of arachidonic acid, and so they have very controlled physiological effects because they bind to receptors and produce all sorts of fascinating physiological effects. But isoprostanes form from random free radical attack on membranes, and the fascinating thing is that people found that there's often 10 times higher levels of isoprostanes floating around in your biofluids in your blood than prostaglandins. So that just tells you how significant some of this free radical damage by endogenous oxidants is, that it's, in quantitative terms, much more prevalent than enzymatic cyclooxygenase dependent metabolism of arachidonic acid. So these products are not toxic, thankfully, but they do have potentially vasoactive effects on the kidneys. Kidneys are vulnerable to prostaglandins as well, and isoprostanes have similar effects on vascular function in the kidney. But these have turned out to be really useful markers of lipid peroxidation, and here's again an old paper from Vanderbilt where they looked at the levels of malondialdehyde in the liver of rats. So they dosed them with carbon tetrachloride, fairly high dose, pretty massive dose. But you could see a dose that increased malondialdehyde only about a few fold. We're going to be measuring malondialdehyde today in your labs if you're doing your lab today. But look at the levels of isoprostanes, gone up 80-fold compared to control. So it's a much more sensitive biomarker than malondialdehyde because it's It's a very specific specific byproduct of lipid peroxidation. So for this reason, people have used isoprostanes for a lot of clinical trials, trying to work out how much oxidative stress is occurring in an individual. So you can look at the onset of disease, coronary heart disease, whether or not drugs are protecting the tissue by looking at isoprostanes. So quite a lot of literature there involving isoprostanes as biomarkers. You can use a variety of different techniques for measuring these species, including mass spectrometry and ELISAs as well. Okay, so now let's just change gears and get to the more interesting products of lipid peroxidation, the toxic ones. And we'll begin with the lipid radicals, and we can sort of jump over these because they're probably not big players in terms of cell damage, but those peroxyl radicals formed during that initiation stage of lipid peroxidation are very important to amplifying membrane damage, but they probably aren't major players in causing cellular injury. They may damage proteins and enzymes. Maybe they react with DNA. We used to work on that for a while, but I don't think they're especially reactive. These peroxyl radicals are also targets for chain breaking antioxidants, and that includes phenolics and vitamin E. But certainly there's lots of them produced. I won't go into this in too much detail, but this paper looked at the yield of lipid radicals in actual cells, and they showed that if you pre-treated in cultured cells with different types of fatty acids and you made the cells richer and richer in polyunsaturated fatty acids, you can measure that by reporting what you call the methylene bridge index. You can control that by loading the cells with different types of lipids, and you can see there's this clear relationship between how many polyunsaturated fatty acids are in a cell and the rate of radical formation, lipid radical formation. Okay, so there's no doubt that these are clear products of oxidative damage to cell lipids, but they're probably not really important in terms of mediators of tissue injury. But then we come to the reactive carbonyls, and these guys really are probably very significant mediators of oxidative cell injury. So they have the ability, after being formed in the lipid membranes, of getting out of the membrane and swimming through the cell and even into adjacent cells and causing toxicity in adjacent cells. And this can be an issue because sometimes, within a complex tissue, different types of cells have different capabilities to metabolize chemicals. So sometimes these sorts of toxic species might be damaging, say, a neuron, which may not have great capability to metabolize aldehydes and lipid peroxidation products. So it can be formed in one cell type, diffuse, cause into another vicinity of another cell type and damage it if that cell type doesn't have metabolic capabilities towards the chemical. Okay, the one we're going to be looking at today in our lab is malondialdehyde, very widely used marker of lipid peroxidation. Today we're going to use this assay, the T-bars assay. We use a reagent thiobarbituric acid, which of course reacts with malondialdehyde as well as other products. Malondialdehyde has complicated chemistry for a molecule that just has 3 carbons and 2 oxygens attached to it. In solution, it equilibrates with an analogue called beta-hydroxyacrolein, and this is also deprotonated. So it exists as what we call an enolate at physiological pH. Okay, so these molecules can react with DNA and protein. They've also been shown to have weak mutagenic activity and activity even in rats in terms of cancer development. Probably more significant in terms of their inherent toxicology would be some of these longer molecules, so longer fragments. So malondialdehyde is just a 3 carbon, it's a small fragment of a lipid, oxidized lipid undergoing breakdown. If you have a larger fragment in the form of 4-hydroxynonenal and 4-oxononenal, non-enol. These are very nasty species indeed. These are very, very chemically reactive, highly reactive with proteins and DNA. They've been implicated in a whole lot of diseases, Alzheimer's disease, chronic heart disease, alcoholic liver disease. We're going to come back to in a minute. And there are interesting differences between them. If you remember my lectures last semester, we distinguished between hard and electrophiles that attack DNA preferentially and soft electrophiles that attack protein. Even though these molecules are very similar, HNE's just got a hydroxy group on the 4 carbon, whereas ONE's got this carbonyl group. It changes their hardness and softness. So HNE is a soft electrophile, reacts with proteins and DNA to a lesser degree, whereas oxynonenal is a hard electrophile that damages DNA readily. Okay, so very nasty species, and it's due to the presence of this double bond in close proximity to the carbonyl group. That's called an alpha-beta unsaturated tryptophan, and all chemists will be very familiar with them because they're very electrophilic. Now, the aldehydes that I've been particularly interested in, and we're going to look at in next week's lab, are the short chain 2-alkenols, and these include things like acrolein, the shortest one with just 3 carbons, and it's the simplest alpha-beta unsaturated aldehyde where you've got a carbonyl group directly adjacent to a double bond on the alpha and beta carbons. This is a highly reactive substance, very nasty substance. Some indication of how nasty this substance is back in World War I. The Allied and Axis powers seriously tried to turn this into a chemical warfare agent. So, like mustard gas and chlorine gas, tried to put it in canisters and fire it at each other. Not a good idea because you can't always control the dispersion of chemically reactive substances in the air. The wind might change and your own troops get poisoned. So that's the problem with chemical warfare. But it also polymerises acrolein, so they didn't know how to stop that in the old days. So thankfully it wasn't used as much as some of the other horrible substances that were used in World War I. But more recently, of course, we've found this molecule is present in huge quantities in tobacco smoke, one of the most prevalent toxic substances in tobacco smoke. But it also forms endogenously during lipid peroxidation, and that's why we're interested in it in our labs next week. Okay, so why are these substances so nasty? Well, they attack DNA, form adducts, so you can see it's guanine, deoxyguanine, and it's got this, in the case of a reaction with acrolein, we've got this new 3-carbon ring added onto the end of the guanine molecule. MDA, malondialdehyde, forms with a hydroxy group attached to it. In the case of acrolein, it's called a propanohydroxy ring. But this, in the case of malondialdehyde, forms this unsubstituted ring. Okay, so these have been found in the genome and they arise endogenously just due to normal production of these substances. Probably more significant from an acute pathology point of view is the fact that these molecules can attack proteins. And this is just an example of malondialdehyde attacking lysine. So this carbonyl group forms a lysine adduct. And this is what we're going to be using as the basis for our assay next week. We're going to screen our compounds to see whether they stop this sort of protein damage by acrolein rather than malondialdehyde. Acrolein is more reactive with proteins and MDA, so that's what we'll be doing next week. These sorts of aldehydes have been implicated in all sorts of diseases, and I just pulled this paper out because I thought it was quite interesting and visually convincing. These folks looked at the levels of protein damage in semen collected from fertile versus infertile males, and look at the ones from the 3 infertile individuals compared to the fertile individuals. Far higher levels of adducts formed by, I think this is 4-hydroxynonenal, so 4-HNE modified protein. So you can buy antibodies that detect adducts formed by these aldehydes on proteins. So this is just a suggestion that this process of lipid peroxidation generating these nasty electrophilic species might be contributing to pretty significant human health issues. Now, because there's such a soup of aldehydes formed during lipid peroxidation, and this diagram here from a recent study highlights about 15 or 20 of them, I think. Acrylene's in there, but you can see some of these other aldehydes as well. And you can see they can attack several different nucleophilic amino acids, lysine, cysteine and histidine, and form adducts. And it's the presence of these damaged proteins within cells that has given rise to this new field of science known as adductomics. So adductomics, just to confuse you, goes beyond the proteome, study of the proteome, to ask what are the proteins in the proteome that are actually carrying adducts formed by some of these nasty reactive species. So the technology that lets us measure all these products and work out what's going on. We saw how it was applied last week in Alzheimer's disease. This is being done to just find out which of these damaging species are causing protein damage in diseases. And then there's another product of carbonyl stress that also are getting more attention, and these are really nasty species. So they're similar to isoprostanes in that the lipid isn't breaking up, it's not releasing fragments, but the whole lipid is oxidised. And you can see this proximity of these two carbonyl groups, sometimes called gamma carbonyls or isoketols. These carbonyls are on gamma separated carbons. These are really nasty products, very reactive, and they attack proteins to form on a nitrogen to form these new 5-membered rings called pyrrole adducts. These have been implicated with just about every nasty degenerative disease known to science, so they really are pretty nasty species. Okay, so just to wrap up, and I won't go through this in too much detail because we're out of time, but one disease where people are thinking these species may be well contributing is alcoholic liver disease. Of course, alcoholic liver disease occurs in relatively small numbers of people that are vulnerable to this condition, but usually in heavy drinkers. The earliest signs of alcoholic liver disease is a fatty stage, alcoholic fatty liver. And so this is associated with heavy drinking, binge drinking, that sort of thing. And for a long time we didn't realise, or there are a lot of theories about what's going on in alcoholic liver disease, because if you keep drinking heavily, it starts progressing to this nasty inflammatory condition, similar to a sort of viral hepatitis, steatohepatitis, so fatty liver with lots of immune cell infiltration, and then all the way through to a very horrible condition known as cirrhosis, alcohol-related liver cirrhosis, where basically a lot of the hepatocytes have died off and have been replaced with fibrotic scar tissue. Really nasty, horrible condition. You don't want to get cirrhotic liver disease. Horrible way to go. And then a few people even progress to, if that's not bad enough, to carcinoma of the liver, hepatocellular carcinoma. Now, for a long time, people thought that the early events here involving the deposition of fatty liver, or formation of fatty liver, was due to events outside the liver, and you get mobilisation of fat in heavy drinkers from other tissues, and it seems to be accumulating in the liver. But then one of the mechanisms that people have found in more recent times is that you're actually getting a lot of damage by these sorts of aldehydes in animal models of alcoholic liver disease. And I won't go through all this, but people found that you can identify adducts on proteins using that biotin capture technology I talked about in my last lecture, where you treat proteins recovered from a tissue with biotin hydrazide and see what you can fish out, pull out of the protein mix, and very often they've found aldehyde adducts formed by these nasty products of lipid peroxidation. Okay, and when you looked at what are the proteins that are getting damaged by these nasties, whole lot of proteins involved in fatty acid metabolism in the liver are getting screwed up and damaged, adducted. So this is one of the reasons why people are thinking maybe these aldehydes are actually doing a bit of damage in heavy drinkers. And also the role of acrolein got a bit of a boost just a few years ago because people began measuring levels of acrolein-derived metabolites. And acrolein is a nasty electrophilic species, undergoes the same sort of metabolism I identified last semester for reactive chemicals. Remember, they get trapped by glutathione and then processed by enzymes in the kidneys. And they appear in urine as mercapturates or mercapturic acids. And you can measure acrolein-derived mercapturate in urine. And it's there in all of us. And depends on what you like to eat. If you like eating chips, potato chips and fried food, your levels of HPMA screwed up after a meal. But lots of it are formed in people with alcoholic liver disease, and it seems to be a good marker of a pretty nasty advanced form of the condition, acute alcoholic hepatitis. You can discriminate between severe and non-severe forms of the disease by measuring this urinary metabolite of acrolein, and also it correlates nicely with some markers of apoptosis such as keratin, cytokeratin 18, as well as some inflammatory markers. Okay, so this sort of research is getting a lot of attention, particularly in the US, and people are looking at acrolein and related aldehydes as mediators of some pretty important diseases. So that's why we want to screen our compound library to see whether it can trap any of those molecules in your library, can trap some of these nasty aldehydes. So this is my main research interest these days, trying to trap aldehydes rather than free radicals. Okay, sorry I've gone a bit long, but hopefully that gives you a bit of an introduction to the background to our labs these next couple of weeks and why lipid